Okay, going to throw a couple more things onto the inhibition thing. So an inhibitor can stick onto the enzyme in a particular spot. I've mentioned that's called the regulatory site. It may be inside the active site, it may be next to the active site, it may be somewhere totally different, changes the whole shape. I also mentioned that a lot of times you have a metabolic pathway where you've got a whole bunch of steps between where you start and what you're trying to get to. Each one of those steps is going to have its own enzyme. And so way down at the end, there's an end product. Now, on some stuff that you need to kind of keep making through the course of whenever, the dent, the regulatory site dent on the very first enzyme fits the end product, the thing that's being made at the very end, but it fits very loosely on there. So think about this. Here's your starting point, here's your first enzyme. Step, 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 step. Make the end product. As this stuff accumulates, it sticks to the first enzyme and shuts it off. It doesn't stick on there really tight, so it sticks on. The more end product you have, the more time the first enzyme is shut down, which shuts down the whole pathway and shuts down the production of that end product. So it controls how fast the whole system works. Because as soon as you build up a certain amount of end product, it's kind of slowing down its own production. So it never gets completely out of control how much you're making. And it doesn't need to shut down all the rest of the steps. It just needs to shut down that first step. And then the substrate is waiting to change because it can't fit into the enzyme to change. So it's a pretty efficient system on a lot of stuff that is made fairly regularly inside the cell, but you don't want a huge buildup of the stuff. Another thing about inhibition, if you're going to turn the enzyme off and then turn it on again, you want reversible inhibition. You want to be able to get the inhibitor off. In end product, it's on there loosely, so it's a reversible inhibition. It's on there, it's not working. It pops off, it's working. It's on there, it's not working. And there's a lot of enzymes where it'll be a more permanent, but reversible, where, okay, we're done with you for a while. Here's the inhibitor stuck on there. You're not going to work until we peel that off again. But we will peel it off again. And sometimes it's like, okay, we're done with you, enzyme. We are not going to be using you in the foreseeable future. We're going to recycle you. Let's stick an inhibitor on there and just shut it off permanently. Irreversible inhibition. And in that case, you'll put it into a peroxisome and break it down and use the bits and pieces to do other stuff. Poisons often do that also, where a toxin will work because it will shut off one of your critical enzymes, and it doesn't want them to turn back on again, whatever's poisoning you. It could be like a mushroom toxin, or a plant defensive toxin, or a venom that something is injected into you, an insect or a spider or a snake. But there's a lot of inhibitors that will shut down something so you can't breathe, or in the case of mushroom poisons, you can't make RNA. That's a really nasty one. That affects everything all over the place. But it's an irreversible inhibitor. So sometimes we do that on purpose, and sometimes other things do those things to us on purpose. Okay, the next thing involves a graph. I hope you can read a graph. This has to do with the effects of temperature on enzymes. How do you measure how an enzyme is working? Well, you measure how fast the reaction it's controlling goes. The better the enzymes work, the faster the reaction rate. Now, very often on a graph like this, you put arrows down because you're not actually going to put little measurements here. That way, this is more of a generic fit, all one size fits all sort of graph. Now, the one I'm going to put up here doesn't fit all because I'm going to make it a little bit too perfect. So at a very low temperature, the particles are barely moving. They're barely bopping into one another, enzyme or not. So it's not working too well. And then as you make it warmer, things are hitting each other. Things are getting into the enzyme. It's working really well. You hit a point here, and I'll come back and talk about this. And there's a point where it's working as well as it can work. The enzymes are grabbing and doing, and they're zipping around and bopping into one another. And then it gets a little bit too warm, and it starts to shake the enzymes apart. Well, not apart. Remember, an enzyme is a big blob with a particular shape for its active site. Heat doesn't just move stuff around real fast. It takes big molecules and it shakes them. And what holds proteins together are hydrogen bonds, which are not necessarily super strong. And if you shake it, some of the enzymes will lose their shape and stop working. So this is what's happening. Too cold, nothing's barely any movement, things are not happening. As it gets warmer, more and more substrates are hitting the enzymes, and it's making the reaction speed up. And it's making the reaction speed up, and it makes the reaction speed up. And this is about as good as it's going to get. Because as you get it warmer, the enzymes some of the enzymes start to change shape and not work. And then more of them change shape and not work. And more of them change shape because it's getting warmer and warmer and warmer. Until eventually, it's like the enzymes aren't working anymore. And you get a rate similar to what you started out with. Now, this is a very even curve. It doesn't always work that evenly, but this kind of gives you the idea. Now, on the temperature scale, that temperature is the temperature at which your enzyme is working its very, very best. That's called the optimal or the optimum. Different books call it either. Gee, you want to guess what the optimal temperature for human beings is? Somewhere in the 97 to 99 range for Fahrenheit. Is that our curve is actually very narrow. It drops out really quickly down around 94 or 95. It drops off really quickly up around 100. That's why you run too bad a fever. Things in you stop working. You cook your brain, that sort of thing. But that's part of what makes it worthwhile to burn all the energy we need to burn in order to keep our temperature very stable. Because almost all of our enzymes, their optimal temperature is our body temperature. So they work super, super efficiently as long as we keep that temperature up. And I will tell you an exception. There's a critical enzyme in sperm production that sperm production, the glands were on the surface at some point in the distant past. And so they were exposed to the environment. And while all the other enzymes were kind of evolving into this one optimal temperature, we were selecting for the ones that could, maybe were a little bit cooler, but there were variants that worked better at this temperature. Some that were maybe a little bit warmer and worked better at that temperature. It turns out that, because in evolution you don't get what makes sense, you get what you get, is to produce sperm, which is really important to make offspring, is that the critical enzyme, we didn't pick one at normal body temperature. What we did was we took the sperm production site and we moved it farther out into the environment where we could keep it cooler. And so that's why the testes in mammals are in that little sac hanging off the body. The mammals that don't have them in the little sac actually have this very complex circulation system that cools them inside the body. But the optimal temperature for sperm production is, I think, somewhere around 92 to 94 degrees. So a decent, temperature below. And if you get too warm, you don't make sperm, or at least you don't make them very efficiently. That's why a really high temperature can actually sterilize a guy. It can actually shut the system down kind of permanently. So what if you don't have your own set body temperature? This is what you sometimes get if you don't generate your own heat, but you use the heat from the environment. These are two enzymes doing the same thing. This is probably burrow temperature for using the enzyme at night when you're hiding in your burrow. This is daytime temperature for using the enzyme in the daytime when you're out and about. So you get something like a snake that has to come out and sun itself, or a lizard that has to sit on a rock and warm up. What it's doing is it's shifting control of this system from the nighttime enzyme to the daytime enzyme, which means it's got two enzymes doing the same thing. And in between, it's not terribly efficient at doing what it needs to do. It has, you go, oh, it's sluggish, it hasn't warmed up enough. Well, it was moving around in the evening, oh, fine. But now it's kind of in this trough between the daytime optimal temperature and the nighttime optimal temperature. So it doesn't take as much energy to do what it needs to do. It's not generating heat, but it has another issue. Things like this need lots of oxygen, lots of food to generate enough ATP energy to keep our heat going. That might be a typical enzyme curve for a plant, like a tree around here, where during the spring and summer, it's got to be able to work at a wide range of temperatures. So the enzymes are working pretty much all the time. They never work super, super great, but it's a plant. It doesn't need to crank a lot of stuff out. But it has an enzyme activity from, when things thaw out in the spring to when it's really, really hot in the summertime. And kind of this range is the mid-range that it's most of the year when it's active, right? Wintertime, it might be shut down. That would be maybe, way down here. So optimals change, the widths of the curves change, the height of the curve change, whether you need one or two, all of that stuff is affected by this stuff. Now this is going to look very, very similar. It's the same basic idea. Oh, let me write this down. When people discovered that you could mess with proteins and get them not to work, they would lose their nature. So they called that disnaturing. And what we now know is anything that changes the working shape of the protein so it doesn't work, prions do that too, remember? They change working proteins into prions, so they're not doing what they were supposed to do. But that's where the denaturing is. Heat can do this. What we're about to talk about can do this. All sorts of different things can denature the protein. An irreversible inhibitor that changes the shape in a way where it doesn't work it will denature it. But it's all shape changes. So you get a very similar curve, except they tend to be very narrow. So you got rate over here, and down here, you have pH. Think back. Proportion of hydrogen, acidity and basicity, acid, base. And this also would have an optimal. For our chemistry, the optimal is very slightly basic, as our tissues tend to be very slightly basic. So our optimal is between 7 and 8. But for some things that live in somewhat acidic environments, the optimal pH may be low. We have bacteria that are active in our stomachs. Their optimal pH is way down around 3, I think. And when they go into your intestines and the pH changes, because the optimal pH for most of your digestion is around between 7 and 8. those enzymes need to be able to work. And as soon as the stomach enzyme is in the pH range of your intestine, it just loses its shape and stops working. What's going on here? Well, pH has to do with hydrogen ions and hydroxide ions. H plus on the low end of the scale, OH minus on the upper end of the scale. And in both cases here, these are very tiny charged particles that can get inside a protein. Proteins are big. There's a lot of space in them. And if there's a whole bunch of positive charges wandering around inside, messing with the hydrogen bonds, it's going to change the shape of the protein. And so a protein will be constructed to be stable with just the right mixture of hydrogens and hydroxides. And if you shift toward more hydrogens, they're going to get in there and mess with the bonds, and the shape is going to change. Too many hydroxides, they're going to get in there and mess with the bonds, and the shapes will change. So both sides of this are denaturing the proteins. Oh, I also should have mentioned this. Back to temperature. As the temperature drops, the screen's reversed. Temperature drops, the enzymes actually constrict a bit. And that makes the fit in the active sites a little tighter, and that also slows the reaction down. It's not as easy to fit the substrates in, and when they change, it's not as easy to get rid of the products. So there's another reason, not just because the particles are all slowed, but the enzymes themselves are not quite as efficient as you cool them off, because the big molecules, like anything, it gets slightly more dense when everything isn't moving quite as much. That's okay, most books don't mention that anyway. Okay, one more thing to talk about. I'm going to stick that down here. Enzymes very often need helpers. I'm going to write this down and then I'm going to talk about it. Leave yourself room. Proteins and enzymes, chemistry very often means moving electrons around, right? Think back back to the first thing when we first started talking about this, and it was all electrons on the outside and going here and going there and trading off and swapping and sharing. Proteins are not great at moving electrons. They often need help. And cofactors are generally minerals, metal ions, metal molecules, things that are a little bit better at moving electrons. And they come in and help the enzymes out. In some cases, they become part of the enzymes. In some cases, they just come in and kind of stick there temporarily, go, yeah, I'll come in and help you out. Okay, I'm leaving again. And it's a way of turning the enzymes on and off. But cofactors are generally minerals. Coenzymes are little bitty molecules of all sorts of different types, but they also very often are good at moving electrons. And there's other things that coenzymes do also. So for the enzyme to work, it needs its buddies. Cofactors in the case of some enzymes, coenzymes in the case of others. Some enzymes work fine all on their own. Now here's a term that goes with this. It goes with coenzymes. There's a long list of coenzymes that we need for some of our enzymes to work. A lot of coenzymes we make, but some coenzymes we also can get in our food. They're little bitty molecules. You absorb them without breaking them down, and they can go to work. Well, evolution is, lazy. Don't make stuff if you're getting it in already made. And what will happen is, even if a coenzyme is something we needed to make in the distant past, if we've been eating it in our food for a very, very long time, we never need to make it ourselves, and the genes for the proteins to make these coenzymes, they stop being useful. And if they mutate to a point where they don't work anymore, what does it matter? This is a very human-centered term. Vitamins are coenzymes that humans need in their food. We cannot make them ourselves. And it should be a very human-centered list. It often is not used that way. But you can take a vitamin, like say vitamin C. Your cat doesn't need vitamin C in its diet. Cats, historically, evolutionarily, not a lot of sources of vitamin C in our diet. Our ancestors ate fruits and leaves and got vitamin C. We need it because we can't make it. Cats have a gene for an enzyme to make vitamin C for them, to make that coenzyme. If you look through our genes, you could find that gene in human genetics, except it's so degraded. It's been mutating and useless for who knows how many generations, because we don't need it. So it's just a weird thing. The vitamins are specifically a list. Now, we're omnivores, so we get a lot of coenzymes from animal foods and from plant foods. So the list of vitamins that humans need is longer than the list of vitamins that a cat needs, or longer than a list of vitamins that a dog needs, although dogs, having been eating human food for a really long time, their list is getting closer to ours. But it always drives me crazy when you see these cat and dog food things with all these fruits and vegetables on them, and they talk about all the wonderful. They don't need that. Is that they're making those vitamins on their own. Anyway, that is the chapter.